Welcome back to the Deep Dive. Today we're shifting gears just a little bit. Usually we take a big messy topic and unravel it for everyone, but today we are going into full-on study buddy mode. We know a lot of you listening are staring down the barrel of an exam on regulatory guidelines, and there's one document that it seems to trip people up because it sits at this really uncomfortable intersection of biology, hard law, and, ethics. We're talking about ICH S11.
That's the one. It's the guideline for nonclinical safety testing in support of development of pediatric pharmaceuticals. It is a mouthful of a title, but it's arguably one of the most intellectually demanding safety nets in modern medicine.
And the stakes here are just, they're different, right? I think the best way to hook everyone in before we even get to the rules is to just state the reality. If you miss a safety signal in an adult, that's bad, potentially tragic.
Of course.
But the philosophy behind S11, it implies that getting it wrong in a child is, well, it's just biologically worse.
It is worse, and not just emotionally, but biologically. I mean, think about it this way. With an adult, you're dealing with a finished product, generally speaking. If a drug is toxic to an adult liver, you're damaging a structure that's already built.
Right. It might heal, it might scar, but it's there.
Exactly. But with children, you're intervening in a system that is literally under construction.
You're not just damaging the house, you're messing with the blueprints while the contractors are still pouring the concrete.
That's a perfect analogy. If a drug interferes with the developmental process, say myelination of the brain or kidney formation, you aren't just causing a temporary injury, you could be permanently altering the trajectory of that organ for the rest of that child's life.
You could change the baseline.
You change the baseline. And that's why S11 exists, to stop that from happening.
Which is terrifying, but also why this is so crucial to master. So here's our mission for this deep dive. We're going to deconstruct the ICH S11 guideline specifically for you, for students. We're not going to read the PDF to you.
No, please don't.
You can do that on your own time. We're going to synthesize the core logic so you can answer the why and how questions on your test.
And we have a roadmap. We'll start with the rules of the road, what this document is, who it applies to. Then we'll spend a good chunk of time on the weight of evidence, or WOE approach. That's the absolute brain of this guideline. I've even got a mnemonic for you.
Ooh, we love a good mnemonic.
Then we'll get into designing the actual Juvenile Animal Study, or JAS, including the rat versus monkey debate. And we'll finish with some common pitfalls, the trick questions that always seem to show up.
Okay, let's dive in. Open your mental textbooks to the definitions page. First thing.
Yeah.
What exactly is this document?
So ICH is the International Council for Harmonization. The whole goal is to get regulators from the US, Europe, Japan, and other places to agree on one standard.
So companies don't have to run different tests for every single country.
Exactly. And S11 specifically covers non-clinical, so animal studies for drugs intended for kids. The goal is twofold. Facilitate safe clinical trials, but also adhere strictly to the 3 Rs.
The 3 Rs.
Reduce, replace, and refine animal use. The guideline is very, very explicit. Do not use animals unless you absolutely have to.
Right. And a crucial exam question is always about scope. What's in and what's out. It's not for everything.
It's pretty distinct. In scope, small molecules and biotechnology-derived pharmaceuticals, what we usually just call biologics.
Okay.
Out of scope. Vaccines, gene therapies, cellular therapies, tissue-engineered products.
So if I get a case study about a new mRNA vaccine for toddlers and I start citing S11...
You're likely failing that question. Yeah. Vaccines have their own set of guidelines. S11 is not the tool for that job.
Got it. Now, before we get to the methodology, we have to talk about the mini-adult fallacy. I feel like this is the philosophical core of the entire document.
It really is. For so long, the lazy assumption in pharmacology was kids are just small adults. You have a dose for a 70-kilogram man, you just do some math, scale it down for a 10-kilogram child, and you're good.
And S11 just completely rejects that. Completely.
Because the biology just doesn't work that way. A child's body is a dynamic, moving target. Their liver enzymes might be working at 150% of adult capacity, or 10%, depending on their age. Their blood-brain barrier is leakier.
And I've read this cuts both ways. Sometimes young animals are more sensitive, but sometimes they're actually less sensitive.
That's true, and it is counterintuitive. Sometimes the receptor a drug targets just hasn't been expressed yet, so you give the drug and nothing happens. No efficacy, but also no toxicity. Other times they're hypersensitive because they can't clear the drug. The point S11 makes is you cannot assume, you have to look.
Which brings us to the engine of this thing. If you take one thing away from this episode, it should be this concept. the wheat of evidence, or WOE.
Absolutely. The S11 guideline does not say if you have a pediatric drug, you must do a juvenile animal study. That is a surefire way to fail your exam.
Okay. So what does it say?
It mandates a thinking process. You have to build a case. You look at all the data you have, and you weigh whether a new animal study is actually going to tell you something you don't already know.
So you have to prove the study is necessary, not just do it to check a box.
Correct. It is all about filling a gap in knowledge. If there's no gap, there's no study.
And there's a specific figure in the document, Figure 1, that lists all these factors you have to weigh. To help everyone remember, we've sort of hacked together a mnemonic. We're calling it the PADS method. PADS. Let's walk through it.
I like it. Okay, PP stands for pharmacology. You have to ask, does this drug target a system that is currently under construction in the child?
Okay, give me a concrete example of that.
Let's say you have a drug that affects bone density.
Mm-hmm.
If you're giving it to a 40-year-old, their bones are formed. You're worried about maintenance. But if you're giving it to a 2-year-old, their bones are rapidly growing.
Ah, okay. The pharmacology itself suggests a very specific developmental risk.
It does. And you also have to look at off-target effects. Does it accidentally hit something else involved in development?
Okay, so P is pharmacology. A is for age.
And this is, I would argue, the most heavily weighted factor in the whole document. It's the youngest intended patient age. And the rule of thumb is simple. The younger the patient, the higher the risk.
And the more likely you need a study.
Much more likely, because the developmental velocity is higher. A 16-year-old is biologically very similar to an adult. But a neonate, a baby under 1 month old, is a physiological alien compared to an adult. Their systems are still building.
So if your drug is for neonates, the bar to avoid a study is incredibly high.
Incredibly high. So if the exam asks which factor drives the need for a study the most, look for the age of the patient.
Got it. Okay, D is for data.
Specifically, the amount and type of existing data. You're not operating in a vacuum? Do you have adult tox data? Do you have clinical data, maybe from similar drugs?
So if I already know from an adult rat study that the drug destroys the kidneys, and I know kids' kidneys work the same way,
You do not need a juvenile study to prove it again. That would be unethical. You already know the hazard. The JAS is only for new questions that the adult data just can't answer.
and finally S.
S is for specificity and selectivity. Is the drug a magic bullet that only hits one target? Or is it a dirty drug that hits lots of things?
Hmm?
Highly specific drugs are often more predictable. If it's less specific, what we sometimes call promiscuous, you have a lot more worry about unexpected side effects.
So PIS, pharmacology, age, data, specificity. If you weigh those and the scale tips toward we don't know enough, then and only then do you design a juvenile animal study, a JAS.
Correct. And that leads us to the next big section, designing that JAS.
And I mentioned students think there's like a standard template. Take 50 rats, dose them for 30 days, check the liver.
And they would be wrong. S11 really emphasizes a customized design. It's fit for purpose. You're only running this study to answer the specific safety questions that your weight of evidence assessment raised.
So if you're worried about the immune system, you design a study that tests the immune system.
Exactly. You don't just run a generic tox screen.
Okay, let's talk about the animals. If we have to test, who are we testing on?
The gold standard is the rat.
Why the rat? Why not something closer to us?
Well, a few reasons. One, we have mountains of historical data on rats. We know exactly how they grow. Two, they grow incredibly fast. You can cover infancy, childhood, and adolescence in a matter of weeks.
Wow.
And third, practically, you can test almost all the organ systems in a rat.
But what if it's a really complex drug, like a biologic? Wouldn't we want to use a monkey, a non-human primate, an NHP?
This is a classic trap. Students often assume primate equals better, but S11 actually discourages using NHPs for pre-weaning studies.
Pre-weaning. Why? I mean, I would think a baby monkey is a much better model for a human infant than a baby rat.
In theory, yes. In practice, it's a nightmare. And it all comes down to maternal bonding.
Okay, elaborate on that.
So, infant monkeys are incredibly dependent on their mothers. If you are trying to dose that infant every day, you often have to physically separate it from the mother or sedate the mother. That causes immense stress.
We're talking cortisol spikes.
Massive cortisol spikes. Physiological stress. I mean, huge changes to the immune system. If you run a study like that and you see the infant monkey has a suppressed immune system, you don't know if it's the drug.
Or the trauma of the study itself.
Exactly. The data becomes confounded. It's useless.
That's a really important practical nuance. It's not just biology. Its feasibility. So unless it's totally unavoidable, stick to rodents.
Rodents or sometimes mini pigs, yes.
What about timing? You said rats grow fast. So if my drug is for a human infant, I can't just dose a rat infant for the same number of days, can I?
No, absolutely not. Their developmental clocks are totally different. It is not a one-to-one ratio. You have to match the developmental window.
And what does that mean in practice?
Let's use the brain. In humans, the brain growth spurt happens around birth and continues for the first few years. In rats, that same biological process happens mostly after birth, in the first 3 weeks.
Ah.
So if your drug is for a human neonate, you start dosing the rat at postnatal day 1 or PND1 through PND 21 to cover that window.
So you're matching the maturity of the organs, not the calendar days.
Precisely. And often the duration in the animal actually needs to be longer than the human clinical trial, just to be sure you cover that entire window of vulnerability.
I want to move into the practical application section of our deep dive. Let's run through a couple of scenarios. Imagine I'm a student, and I get this case. Drug X is for adolescents, age 12 and up. We have full adult rat tox data. The drug targets a stable metabolic pathway.
Yeah.
Does this trigger a JAS?
Okay, if I'm applying PAYS, I'd look at age first. Adolescence. Biologically, a 12-year-old is remarkably similar to an adult. Organs are mostly mature. We have adult rat data. That's the D. So unless there's a very specific concern about puberty, the weight of evidence would likely say, no JAS required. The adult data is good enough.
Okay, scenario B. Drug Y is for premature neonates babies under 1 month old. It's for chronic use. And the drug targets a receptor involved in kidney development.
Red flashing lights everywhere. Age, neonate high risk, pharmacology, targets a developing organ. Data. Adult data won't tell us enough because adult kidneys are finished. Verdict. A JAS is absolutely required, and you design it to look specifically at renal endpoints.
That makes the distinction really clear. Now let's talk about growth. This isn't simple, right? Weigh the animal, see if it grows. But you said this is a major pitfall.
It is the most common mistake students make. In these studies, you will often see animals lose weight, but is that developmental toxicity?
I would assume so.
Not necessarily. What if the drug just tastes terrible? The rat eats less and weighs less. That's a nutritional effect, not a direct toxic effect on growth.
So how does S11 say we prove it's a real growth defect?
You have to measure long bone length, usually the femur. If the bone itself isn't getting longer, that is a true growth impairment. If the animal is just skinny but the bones are growing normally, it's likely just a feeding issue.
You have to distinguish between failure to thrive and failure to grow.
You have to. It's a critical distinction for the exam. Bone length, not just the bathroom scale.
Okay, another concept. Reversibility. If a juvenile animal gets hurt by the drug, do we need to prove they get 100% better?
S11 is pretty pragmatic here. You generally include a recovery period after you stop dosing, but the guideline says you don't need to demonstrate full reversibility. You just need to show a trend towards reversibility.
So if the damage is lessening, we can assume it'll probably heal.
That's the idea, but there's a big catch with the brain, right?
The CNS.
Yes. The scary thing about early exposure is delayed effects. You might damage a neural pathway in a baby rat, but you won't see the behavioral result until that rat tries to learn a complex task as a teenager.
It's like a ticking time bomb.
It is. So for CNS drugs, you often have to keep the animals alive for a long time after dosing way into adulthood to run memory and learning tests. You can't just check them the day after the last dose.
Wow. It really shows how complex safety is when you add time and development into it.
It does. And there's one more scenario we should probably touch on. Pediatric first development.
Right. This is when a disease only exists in kids, so you never even test the drug in adults first.
Exactly. You're flying blind. Usually you have adult data to lean on, but if you have a rare genetic disease that only kills babies, well, in that case, the guideline is much stricter. You generally need 2 species, a rodent and a non-rodent, before you can go into humans.
You're basically rebuilding the whole safety package from scratch, but in juvenile animals.
That's a heavy lift, but it's necessary.
Okay, we are coming up on time. Let's do our if you remember nothing else summary. I'll throw the headers at you. You give me the 1-sentence takeaway for the students.
Let's do it.
Point 1, the core philosophy.
WOE is king. Never assume a study is required. Always apply the weight of evidence first.
Point 2, the biggest risk factor.
Age matters most. The youngest intended patient age is the primary driver. Neonates are high risk, adolescents are low risk.
Point 3, the animal model.
The rat rule. The rat is the preferred species. Avoid monkeys for pre-weaning studies because of maternal stress.
Point 4, study design.
Targeted design. JAS designs are fit for purpose, not a checklist. You only include endpoints that match your specific safety concerns.
Point 5. Measuring growth.
Bone length. Body weight is not enough. You must measure the femur to prove a real skeletal growth effect.
And finally, point 6. The goal.
The 3 R's. The whole goal is to facilitate safe trials for kids while minimizing animal use.
Boom. That is your cheat sheet.
I do want to leave the listeners with one last thought, sort of a provocative thought to mull over, specifically on that pediatric first scenario.
Go for it.
We talked about how rigorous this guideline is, but just think about the translation gap. If we have a drug for a condition that only affects human infants, we're relying entirely on a 3-week-old rat to predict the complexity of a human infant's brain. And we do it because it's the best tool we have. It's so much better than nothing. But S11, it implicitly acknowledges that this is an estimation game. The guideline forces us to be rigorous, but it can't magically make a rat brain into a human brain. It just highlights that in pediatric development, there is always this element of the unknown that you just have to manage.
That is a sobering thought, but it really underscores why understanding this guideline is so important. You're not just checking boxes. You're trying to bridge that gap as safely as humanly possible.
Exactly.
All right, everyone. Go back and look at that WOE factors diagram in the PDF Figure 1. If you can visualize that, you can answer almost any question they throw at you. Good luck with the study, and we will see you on the next deep dive.
Good luck.